glycolytic process secretion. So I found this little picture here that kind of represents these three processes. Okay, and in this picture, this would be the glomerulus from its capsule. Here's the renal tubule. So filtration occurs here in the corpuscle, and along the renal tubule, some of the solutes, some of this filtrate gets reabsorbed, other solutes get added. Now, when the final product gets released or excreted into the minor calyx, it becomes urine. So let's look at the filtrate. We said the glomerular filtrate is the fluid and solutes which collect in the capsular space. It's composed of water. Water is going to move freely across the filtration membrane. It's going to carry with it electrolytes, sodium, potassium, chloride, bicarb. Okay, in fact, these electrolytes are going to move freely. Why? Because they're very, very, very tiny particles. Okay? They're small atoms or molecules. They'll be filtered freely. And then we'll also filter out waste products. Some of the principal waste products are urea, creatinine, and uric acid. Where does urea come from? It comes from the metabolism of proteins. Okay? I don't know if you remember this, but all proteins have an amino terminal and a carboxyl terminal. In fact, if we start with an amino acid, every amino acid has an amino terminal which contains nitrogen. It's a nitrogen-containing group. It's got a carboxyl terminal. Now, when our body metabolizes proteins, what happens is oftentimes this amino group gets removed. In biochemistry, they call it deamination. Deamination. And the problem with then the product here, which is ammonia, is that it can be toxic. And so the way the liver handles this is it will take two of these ammonias and link them up with a carbon like this. This product is called urea. Urea is less toxic and is easily excreted. So in the metabolism of proteins, we form ammonia. Ammonia gets incorporated into urea, and urea is a relatively small molecule that then gets excreted. Now sometimes if you're looking at a lab report, they'll call it urea. Sometimes they'll call it BUN. What does BUN mean? Blood urea nitrogen. So if you look in the lab report, they'll mention serum concentrations of various electrolytes, and they'll mention BUN and creatinine, maybe. Okay, if you're doing kind of what they call a mini profile. Okay. Well, BUN is a waste product formed from proteins. Creatinine, what is that? It's another waste product. It's derived from muscle. Every day we break down a very small percentage of our muscle. And when these muscle cells are broken down, creatine gets released. Creatine gets converted to creatinine, and creatinine is excreted in the urine. So creatinine is another waste product derived from the breakdown of muscle. Uric acid is derived from the breakdown of nucleic acids. Not every cell in our body except for red blood cells have nucleic acids. And these nucleic acids are long nucleotide chains. Remember, ASB1. If it's DNA, it's a double chain. If it's RNA, it's usually a single chain. Okay? And these individual nucleotides incorporate nitrogenous bases. You remember what adenine, thymine, cytosine, guanine. Okay. If these nucleotides have incorporated adenine and guanine, okay, then we call them purines. Well, when purines are broken down, you produce uric acid. So, the long and short of it is that uric acid is derived from the breakdown of nucleic acids. So when cells die, and every day we have millions and millions of cells that die, then those nucleic acids are broken down, and when we break down the ones that contain adenine and guanine, then we produce uric acid. Biochemistry, to help you remember that the purines are those that contain adenine and guanine, we say pure as gold. But there's gold. Contact. There's another one. Anyway, that's not. So, 1, 2, 3 waste products. What type of patient, what type of disorder is associated with excessive production of uric acid? Do you know? What disorder is associated with excessive production of uric acid? It's gout.
What are the symptoms of gout? I've heard about it.
Well, there can be many different symptoms. What happens with gout is, and many times it's a metabolic disorder where people produce too much uric acid, it becomes super concentrated within the serum. And so it begins to precipitate out a solution. And it typically will precipitate out within joints. In fact, the most common joint that it precipitates out of is the first metatarsal phalangeal joint, which is at the base of the big toe. And so gout will cause extreme pain and swelling and tenderness. We used to have, Mr. Joseph used to work here. He worked in maintenance, and he had gout. And the pain was so severe that on days when the gout began to flare up, if he came to work, he would wear these shoes where he cut out the toe box because he didn't want anything touching his toe on those days. In fact, the first description of gout back in the late 1800s was by a physician who described the symptoms. And he said the pain was so intense that just the sheet of his bed touching his big toe caused excruciating pain. Okay. Now, these uric acid crystals can also precipitate out within the kidney. And so one type of kidney stone is a uric acid stone. People who have renal insufficiency, whose kidneys don't function normally, okay, these waste products build up. Okay. In fact, we use BUN and creatinine, serum levels of BUN and creatinine, as markers of kidney function. A normal creatinine level in the serum, and it's going to vary some from lab to lab, but it's about 0.6 to 1.2 milligrams per deciliter. Okay? So when you do a chemistry profile where they report the creatinine level, if the creatinine level is elevated above 1.2, if that's the upper limit of normal, then it reflects the fact that that patient has some degree of renal insufficiency. The same thing holds true for BUN, except the normal BUN is somewhere between, let's say, 10 and 20 milligrams per deciliter. Again, it varies depending upon the lab. And so if a patient has renal insufficiency, not only will their creatinine climb, but so will their BUN. Okay, these waste products that are normally excreted by the kidney won't be excreted as efficiently, and so they'll begin to climb. A normal ratio of BUN to creatinine is about 20 to 1. We can take the reciprocal of the creatinine and use that as an index of the percent kidney function that is still remaining in someone who's got renal insufficiency. For example, if someone has a creatinine level of 2, that's elevated, that patient has renal insufficiency. The reciprocal of 2 is 1/2, or 0.5. That would indicate that the patient has 50% residual kidney function. If someone has a creatinine that's climbed up to 4, of course they only have 25% residual kidney function. When someone gets up to 10, we say they have end-stage renal disease. They're not going to survive unless they get either a transplant or dialysis. The reason that we can use creatinine and BUN as markers of kidney function is this, is that for creatinine, we break down a constant percent of our muscle mass every day. And the only way we excrete that creatinine is through the kidneys. So there's an equilibrium that exists between breakdown and excretion. And with that equilibrium in place, our creatinine level remains fairly stable. The only way the creatinine is going to rise is if, number one, your kidneys stop filtering it, the function is decreased, or if you have some type of massive crush injury, okay, where you've damaged a lot more muscle, and so that creatine gets converted to creatinine. Okay. The same thing is true with regard to BUN. You know, we metabolize a fairly constant amount of protein every day, and so it gets excreted by the kidneys. There's a fairly constant relationship between breakdown and excretion. So these are the markers we use for kidney function. Now, uric acid is more variable. Some people have a genetic predisposition to produce more uric acid. So the levels within the bloodstream are going to be more variable from individual to individual. So we can't use that so much as a marker as we can the other two. Now, small organic molecules are also fairly freely excreted, and that would include glucose and amino acids. Larger molecules tend not to get filtered and therefore don't get excreted. So, for the most part, proteins are not filtered, so we don't find proteins within the urine. They're too big. The other thing we talked about the last class is that they tend to have a negative charge, which is repelled by that middle layer of the filtration membrane. When I say glucose is freely filtered, here's what I mean. If somebody has let's say within their bloodstream they have a glucose concentration of 200 milligrams per deciliter. These fenestrations here are going to freely filter water. And because glucose is a relatively small organic molecule, it moves with water. So that its concentration within the filtrate is going to be 200 milligrams per deciliter. In other words, its concentration within the water of the plasma is going to be the same as its concentration within the water of the filtrate. That's what I mean when I say it's freely filtered. There's nothing that slows it down or decreases its movement. So, what determines the rate at which we filter plasma or blood? It's determined by what's called the filtration pressure. GHP, glomerular hydrostatic pressure, capsular hydrostatic pressure, and glomerular oncotic pressure are all factors that affect filtration because they affect filtration pressure. Okay. Glomerular hydrostatic pressure is simply the blood pressure within the glomerular capillaries. Okay? That pressure within the capillaries is a force that favors the outward movement of water and solutes. Okay? So glomerular hydrostatic pressure is just the blood pressure, the mean blood pressure within those capillaries that's filtering fluid, that's pushing it out. Okay? Now, as fluid collects within the capsule here, then we develop a capsular hydrostatic pressure. We cause a buildup of pressure within the capsule that serves as a back pressure that limits filtration. This back pressure is in the opposite direction. So it's one of those factors that affect filtration. And the third factor that affects filtration is glomerular oncotic pressure. That's pressure created by protein within the blood of the glomerulus. These proteins tend to hold on to fluid. So again, this would be a back pressure that opposes filtration. It's drawing fluid into the glomerular capillaries, not pushing it out. So these are the three factors that influence filtration. Now, you'd say, well, what about capsular oncotic pressure? Well, because proteins don't tend to get filtered, you're not going to have any capsular oncotic pressure, typically. So that won't play a role. So here we see the influence of these three factors. Of the three, the only one that's favoring filtration or extravasation of fluid is glomerular hydrostatic pressure. Glomerular oncotic pressure favors the movement of fluid into the bloodstream. Capsular hydrostatic pressure does the same. It opposes filtration. So let's put some numbers to these factors.